Thank you. Thank you all for joining. So my name is Hannelore Aerts. I'm a PhD student in Ghent University with the lab of Daniele Marinetto. And today I will tell you a little bit about a project that I've been working on lately, which involves the development of virtual brain models that might be useful to optimize brain tumor surgery. Okay. So on this figure you can see the brain of a patient that was included in my study who has a tumor in his right hemisphere, so in the right part of his brain. And as you can see in this figure, there are quite some important neural elements that are close to the tumor. So for example, you have some white matter tracts appearing. So these are like the physical connections that exist that connect different brain regions. And to put it with a metaphor, we can think of these as the structural highways, the anatomical highways in the brain. Now, besides the structural connections, we also have regions of functional activation that are close to the tumor. So, for example, here the orange blobs that you see is the motor network. So all of these regions, they light up together in response to a motor task. So to continue with the metaphor, if the structural connections are the anatomical highways of the brain, we can think of the functional connections as the traffic that runs along these roads. So when a neurosurgeon wants to remove such a brain tumor, they will typically rely on this kind of information to perform or plan the surgery. And this is very valuable information, but unfortunately it is insufficient to predict the long-term effects of such surgical interventions. And this is because the brain is a very complex system, which is slightly different for every single patient, and even more so as a result of disease processes, such as the infiltration with the brain tumor. So as a response to that, the brain can change. So we cannot rely on like the average functioning or the functioning of the average brain. And this is why computational modeling could be especially helpful. So computational modeling could be helpful, but how does this work exactly? Okay, so so this figure gives you a summary of how the basic workflow is of computational modeling. And the basic idea behind it is that you start with your neuroimaging data, and on top of this neuroimaging data, you apply some kind of biologically based model of how the brain works in order to simulate brain dynamics. So now in a bit more detail, we start by putting the patient into the scanner and acquiring diffusion-weighted imaging data. We process this data, and based on this information, we can estimate all the white matter tracts, so all the roads that exist in the brain. This information can then be transformed in a more abstract fashion into a structural connectivity matrix or a brain network model. So the information that is captured in both is the same. So, for example, for the network model, the gray spheres, they represent different brain regions, and then you have connections between all of these regions that capture the strength of the structural connection between these regions. In the matrix format, the rows and the columns, they represent the brain regions, and then the matrix entries, they capture the connectivity strength between any of these regions. So once we have this information, we can input it into the virtual brain, which is an open access neuroinformatics platform that allows simulation of brain dynamics. So basically what it does, there are many different types of brain model that you can use, but the ones we use is the reduced Wang-Wang model, but that doesn't really matter so much. So the idea is that you simulate brain dynamics in every single brain region, and then you connect all of the different region models by using the information from your structural connectivity matrix that you applied or supplied as input, weighted by some global parameter. And as output, you can then get time series of brain functioning. So with the virtual brain, it's very practical that you can choose which kind of signal that you would like as output. So you can choose from EEG, MEG, or BOLT time series, and we chose for BOLT time series as output. And then we can transform these signals again in the format of a connectivity matrix, with the only exception now that so the brain regions are still captured in the rows and the columns, but now the matrix entries, they represent the functional connectivity between the regions instead of the structural connectivity. So those who are not familiar with functional connectivity, it means that we compute the similarity between the time courses of different brain regions, for example, using just Pearson correlation. Okay, so this is a general idea of computational modeling. And the first question we asked ourselves is, can we use the same model for brain tumor patients compared to healthy controls? So to answer this question, we also acquired resting state fMRI data here. We pre-processed the data up until the point where we get these region average time series. And then again, we transformed this information into a matrix format, very similar to the simulated functional connectivity, with the only exception now that instead of simulated BOLT time series, we used the actual observed BOLT. And then what we did is we simulated brain dynamics with a whole range of parameter values, and we looked at those parameter values that actually optimize the correspondence between our simulated and the empirical functional connectivity. So those optimized model parameters then represent the parameters that lead to the most biologically plausible brain dynamics. And afterwards, we can then do some statistics on these parameters, examine relationship with other variables, or do some group comparisons. Okay, so this was the framework that we applied in our first study. And so we acquired some data. So we had a sample of 25 brain tumor patients and 11 control subjects. But there were two types of brain tumor patients. So we had 14 meningioma patients. These type of brain tumors, they grow on the outside of the brain in the meninges. And they are generally benign tumors, with the only the thing that they can influence how the brain works once they start growing and exert pressure on the brain. So those are the meningioma patients, and then we had 11 glioma patients. So these tumors, they develop from the glia cells, and they are typically infiltrative in nature and malignant. And then from all of these patients, as well as from the controls, we acquired several measures, among them MRI images, as I already told you. We had diffusion-weighted imaging, but also some anatomical images and resting state fMRI. We also assessed their cognitive performance, and we had some questionnaires to get a little bit of background information on the patients. And then some first results. So the first important thing that we found was that it is actually quite important to optimize or personalize your virtual brain model. So here on the y-axis you can see the correlation between our simulated and empirical functional connectivity matrix. So this is basically a measure of model fit. And then we have 4 different scenarios in which we varied between using control average model parameters, so you can consider these as the default parameters, and individually optimized model parameters. And then we also used the individual structural connectivity matrices, or just an average connectome. And as you can see, the model fit is best in scenario D, where we used both the individual structural connectome as well as individually optimized model parameters. And this is actually quite promising because we would like to apply this framework to improve personalized medicine. So then it's very important then that if you actually personalize your model, you get better results. Now, you can also see that scenario C is also quite good. And in this scenario, we applied, we just used an average structural connectome. And on this average structural connectome, we optimized the model parameters by checking the correspondence with the individual's functional connectivity. So there is still, the model parameters are still individual, but the input of the structure is just a general connectome. And as you can see, the model fit is almost as good as in scenario D. But this is something that has been found in other computational modeling studies as well. And a study dedicated to this problem has shown that it might be related to the fact that the variability in structural connectomes between subjects is much smaller compared to the variability in functional connectivity. So this might be an explanation for this finding. So in the future, once we start making even better structural connectomes with higher resolution data or more high quality data, more fine-grained parcellation schemes, so this could then be possibly improved. But for now, we can remember that it pays off to optimize the model parameters. Then, secondly, we also found that our model parameters, they differ not only between our brain tumor patients and the controls, but also within the brain tumor patients, depending on the distance from the tumor. So here you can see three box plots. So the first one is the group of controls, then in the middle you have the meningiomas and then the glioma patients. And you can see that in regions that are further away from a tumor, the amount of inhibition in these non-tumor regions is much higher compared to levels in a healthy brain. Whereas in regions that are very close to the tumor, we can see the opposite with much lower inhibition compared to healthy levels. And this was actually quite surprising. We didn't expect to find such big results because we didn't specify to the model which were the brain tumor patients, which were the control subjects. We didn't specify where the tumor was located, but still the model could pick up some differences in the dynamics, and we found these results. So, and then further, we found that these model parameters also correlate with structural network topology and to some extent with cognitive performance. So regarding the structural network topology, we mainly found associations with global efficiency of the structural network, especially the association between the global coupling parameter and global efficiency is something that has been demonstrated before in stroke population as well. Regarding the associations with cognitive performance, we find some associations, although you can see on the plot that the relationship is not nicely linear, so we would need some more data to get a better understanding of how this exactly works. So this was a very brief summary of our main findings. If you're interested, you can find some more information in the paper, or you can come to me afterwards. Okay, so I told you that we were going to improve pre-surgical planning. So how can we extend all of this to this purpose? Well, the idea is that we could perform virtual neurosurgery. So if you look closely in this figure, I removed the tumor, and with it you can see that some of the white matter tracts are removed as well. So we can extend this, and using this framework, it might be possible to experiment with different neurosurgical approaches. So to approach the tumor from different angles to try partial resection versus complete resection, or even increasing the resection margin beyond the tumor. We could start even experimenting to what extent, how far we can go with removing possibly tumorous tissue until the network breaks down. Yes, okay, thank you. And for every scenario, we can then reconstruct the structural connectome, run our simulations, and then we can choose from the simulated brain dynamics, we can choose which scenario is the best, which neurosurgical strategy leads to the best outcome. So for this study, we also acquired post-operative data of the same patients. We had a little bit of dropout, which is unfortunate, but still we had some subjects 5 to 11 months after surgery. And then the idea is, as a first step, to repeat exactly the same procedure we did before surgery, to do it on the post-operative data. So that means that we will try to optimize the model parameters and then investigate how these model parameters might have changed over time. How they might have changed after the removal of the tumor, but also how they might have changed in the control subjects, so that we have a clear baseline of what we can expect of normal variability over time. And then in the second step, the idea is that we will try to simulate the actual surgery. So based on we can draw a resection mask after the surgery to get a clear idea of which white matter tracts were removed after the surgery or because of the surgery. And if we then apply this resection mask to our preoperative connectome, we can evaluate exactly, or we can simulate what our model would predict that would be the effects of the surgery. And then we can compare this to the actual post-surgical brain dynamics. And this would be, of course, a very important step to evaluate exactly whether our approach would work or to what extent it might work. But this is still work in progress. So as a take-home message, I would like to emphasize that it is possible and preferable to create these personalized virtual brain models, that the model parameters are sensitive to a presence of a brain tumor, and they relate to structural network topology and maybe to some extent to cognitive performance, and that we are exploring the possibility of using this virtual neurosurgery approach to improve the pre-surgical planning. Okay, so thank you all for your attention, and thank you for the organizers for the opportunity to present my work here. Thank you.
Thanks, Hannelore. We are now in a 5-minute round of questions to the audience in general.
So thanks for this very nice talk. I have a question about the tractography you are using. So I'm not in this field, but from what I understood before is that tractography is suffering from a lot of false positive, no? Mm-hmm. So how can you deal with this problem and how it's affecting the prediction of the surgical model?
Yeah, so that's a very good question. Tractography has come a long way. So in I think the last 10 years we have gone from the very, well, relatively simple diffusion tensor model to now you have all of these constrained spherical deconvolution techniques. So it has improved already a lot, but it's true that there are important biases in tractography, which also impact the generation of the structural connectome that we use as an input. Of course, I think with every technology, you have to start with the best that is available at the specific moment. So we have developed a state-of-the-art pipeline to get the most information out of the data as possible. But it will always improve. Now, I will add to your concern. Especially in brain tumor patients or very close to the tumor, the MRI signal is very likely to be biased. So this is an additional thing that needs some more work, but I think it's you can spend an entire PhD just trying to optimize tractography algorithms to run close to the tumor or even through the tumor if there are some white matter tracts. Because for the moment, once you come close to the tumor, you can see that the signal intensities, they will change. So the tractography algorithm, they will stop. So that's definitely something that should be investigated more thoroughly. Thank you.
Thank you for your talk. I have a question that's quite related to this. So first, to your comment you just mentioned, how do you deal with that problem somehow in the tractography? And another thing I was curious about, how do you define your regions in a brain with such a huge tumor? How did you consider that in the atlas you fit? Or?
Yeah, that's a very good question. Because we spent quite some time, especially on the second part. The first part I can be very short. We don't do anything special regarding the tractography. We run the standard approach, also because for this purpose that we did so far, so the optimization of the modeling parameters and examining whether they are different depending on the distance from the tumor, it doesn't really matter so much that you can get really very close to the tumor. If you're in the area of the tumor, or we are in the area of the tumor, and we already find some differences. Regarding your second question on how to deal with parcellation, that is something entirely different, for which we have created quite a sophisticated pipeline. So for the meningiomas, all of our parcellation schemes, we use FreeSurfer. So FreeSurfer is a semi-automatic procedure based on the Solchi and the gyri of the brain. And in the meningioma patients, it works very well, to our surprise. You can, except for a couple of very large tumors, as the one that I showed you in the beginning, that's very large, then you need to manually edit some things. But in general, FreeSurfer recognizes that these tissues, it's not brain, it's on the outside, as it should be, and it's segmented out of the parcellation. And the other regions are warped correspondingly. So the greater the tumor gets, the more kind of warped your parcellation will be. So that was the first very nice result. For the glioma patients, it's an entirely different scenario, because these tumors, they start growing from inside the brain, and they grow, they infiltrate healthy tissue. So what we did in these in these patients was, first we started by manually segmenting the tumor, and then we ran it in a semi-automatic procedure to optimize the boundaries, and then we gave this tumor mask to a normalization toolbox that is developed in Paris. And this normalization toolbox does is it copies the healthy tissue from the contralateral side into the tumor mask, so that you get a normalized brain, or at least an approximation of how the brain looked before surgery. And this normalized brain was then ran through FreeSurfer to get the parcellation. And afterwards, we just used the lesion brain, but with the parcellation after normalization.
I think we can thank Hannelore once more for an interesting talk. Thank you.